So I'm going to introduce our very first speaker. Dr. Jennifer Juno is a T-cell immunologist with a particular interest in T follicular helper cells and unconventional T cells. Originally from Canada, she was awarded her PhD from the University of Manitoba, where she studied unconventional T-cell dysfunction during HIV infection. Following a 2-year postdoctoral fellowship at the Public Health Agency of Canada, she moved to Melbourne to join the Peter Doherty Institute for Infection and Immunity. Jennifer now leads a team focused on harnessing specific T-cell subsets to improve immunity against human infectious diseases. So it's a great pleasure to welcome Jennifer to give a talk today. And the title of her talk is The Establishment and Recall of SARS-CoV-2 Spike Epitope-Specific CD4 Positive T-cell Memory. So over to you, Jennifer, if you'd like to share your screen.
Great, thank you. Are we sharing properly? Yes, excellent. All right, thanks so much for inviting me to talk today about some of our recent work, and thanks everyone for joining us no matter what time zone you're in. So in the context of SARS-CoV-2 infection, there are a number of different ways in which T cells can contribute to our antiviral immune response. Of course, we know that CD8 T cells can recognize a broad selection of antigens from the virus that will allow them to recognize and eliminate infected cells. And on the other side of things, CD4 T cells can support the CD8 response, secrete antiviral cytokines, potentially exhibit cytotoxic functions themselves. But mostly what we're interested in is their ability to help support the B cell and the serological response. And in particular, we like to study T follicular helper cells, or TFH, which are specialized cells that provide contact-dependent help to B cells in the germinal center. And we know that TFH in the germinal center are really key to supporting B cell maturation, somatic hypermutation, and the establishment of high-quality neutralizing antibody responses. But of course, in the majority of human cohort studies, it can be quite challenging to directly access TFH cells in germinal centers from lymph node samples, particularly in the context of infection. And as a result, we often study a surrogate of these cells in the peripheral blood called circulating TFH, or cTFH cells. These are CD4 T cells with a memory phenotype that express CXCR5. And thanks to a lot of work that's been done in the area of influenza, yellow fever, and other viral infections and vaccinations, we know that the activation of these circulating TFH cells correlates very nicely with the serological response to infection or vaccination. So it was within that context that early on in the first few months of the pandemic, we became interested in trying to understand and characterize how the circulating TFH, as well as the CD4 T cell response, reacts to the spike protein of SARS-CoV-2. And so initially, our studies focused on using an activation-induced marker assay to assess and quantify the cTFH responses to the SARS-CoV-2 spike protein, or RBD. And we recruited a cohort of individuals who had recovered from mild COVID-19 infection. So these individuals were not hospitalized. They had a relatively mild course of illness. And we found that in nearly all of the individuals in our cohort, we saw robust cTFH responses directed towards the SARS-CoV-2 spike protein. And when we took a step back and more comprehensively considered the T cell responses, the B cell responses, and the antibody responses in this cohort, we were able to ask what was the best correlate or predictor of the neutralizing antibody response among these individuals. And we found a couple of interesting observations. First, as we might expect, we found that the frequency of the spike-specific cTFH response correlated positively with the neutralizing antibody titers. And this is really consistent with what we know from other infections and was relatively to be expected. But we also found that there were particular phenotypes of the cTFH response based on the expression of CCR6 and CXCR3 that had either positive or negative correlations with neutralizing antibody titers in this cohort. And this really led us to ask questions as to how the heterogeneity of the cTFH response could influence the subsequent antibody response in COVID-19. So we went on further and we are interested in mapping the spike-specific responses to identify particularly immunogenic epitopes with the hopes that we might be able to study epitope-specific responses over time. And one of the more immunogenic epitopes that we identified was this peptide in the spike-751 position. It's a peptide in the S2 region of the spike protein. And we identified a number of individuals in our cohort with these peptide-specific responses. So our goal at this point was to assess whether we could move away from using the activation-induced marker assay and instead develop tetramer peptide HLA reagents that would allow us to directly track and characterize these cells ex vivo. Now, this can be quite challenging for class 2 tetramers in some cases, but there are a number of studies that have showed how important these reagents can be, particularly for CD8 T cell responses. So the development of tetramers and dextramers have allowed the direct enumeration and characterization of CD8 T cells in convalescent cohorts over time. So with this information, we further went on to try to characterize the restriction of our S751 peptide and to develop tetramer reagents to directly identify these cells. And we found that this S751 response is restricted by HLA DRB115, 1501 or 1502 alleles, and that we were able to generate tetramers that directly identified these cells. We were able to find a very clear population of spike-specific CD4 T cells, individuals carrying the appropriate HLA-DR alleles, which were absent in non-HLA matched individuals. We did a number of QC steps to validate our tetramer. And specifically, one of the nice things that we saw was that the S751 tetramer identified a clear population of cells, which was quite distinct from the S236 epitope, which is another HLA-DRB115 epitope that was identified in mRNA vaccinated individuals. So going ahead and using these reagents, we are then able to assess the establishment of spike-specific T cell responses in our convalescent cohort. What we saw was that in early convalescence, so about 20 to 60 days after the onset of symptoms, we saw a 34-fold expansion of spike-specific CD4 T cells in our convalescent donors compared to an uninfected and unvaccinated control cohort. And among our convalescent subjects, the tetramer-specific cells were almost uniformly CD45RA negative, as we would expect, and they had a very clear central memory phenotype, CCR7, CD27 double positive. And this was quite striking to see, because recently Paul Thomas's group has identified another spike epitope, S167, which is another prominent immunogenic CD4 T cell target. And those cells identified by that tetramer are almost exclusively effector memory cells. So we have two really nice reagents now in the field where we can characterize two quite distinct populations of spike-specific CD4 T cells. But the real question became, what happens to these cells over time after they're established by primary infection? So we were very lucky to be able to track the individuals in this cohort for up to 15 months after their initial infection. And what we found was that after the contraction of the early primary immune response, from about 6 months onward, we saw a marked stabilization in the frequency of our tetramer-specific cells. So between 6 months and 15 months post-infection, frequencies were quite stable with a half-life of 377 days. And again, the frequencies of tetramer-specific cells that we found at these late time points were still about 10-fold higher than what we had found in our uninfected, unvaccinated control cohort. But the real question here was, what was the phenotype of these cells? This represents the bulk CD4 positive tetramer binding population, but what about those cTFH cells that we know are established by infection? And it turns out when we tracked the cTFH specific population, we saw that again, the vast majority of participants maintained detectable frequencies of spike-specific cTFH out to 15 months after their initial infection. So not only were these cTFH cells quite remarkably induced by the infection, but they seemed to be as stable as the rest of the CD4 T cell population, which was really encouraging to see. Now, this sort of addressed the biogenesis of the T cell response in the context of infection, but the next question that we wanted to ask was how this stacks up to vaccination, particularly in the era of widespread vaccination. And so we recruited a second cohort of individuals with HLA-DRB115 alleles who received predominantly mRNA vaccines, but also a few individuals who received adenovirus vaccines or protein-based COVID-19 vaccines. And again, in all of the participants that we studied, we saw a clear induction of S751-specific tetramer response, and this peaked after the second dose. We could start to see proliferation of these spike-specific CD4 T cells as early as day 7 after the first dose, which was actually prior to the induction of the detectable antibody response in this cohort. What again was interesting to see, I think, was that if we compare the frequencies of spike-751 specific cells in early convalescence to the peak frequencies after the second dose in our vaccinated individuals, we saw very similar frequencies between those two cohorts. So infection and vaccination in this particular case seemed to drive similar frequencies of spike-specific CD4 T cells. Now, in the interest of time today, I don't have a lot of chance to go into some of the phenotypic analysis that we've done, but I did want to highlight that there were some differences in the phenotype, particularly around CXCR5 expression of the spike-specific cells that we saw elicited from vaccination and from infection. And I'm happy to talk about that later if anyone has any questions. Now, we followed these individuals in our vaccine cohort for up to about 6 months after their second dose, at which point a number of them elected to receive a third vaccination. So we were able to assess the impact of the booster dose on the T cell response. It's fairly well known now in a number of cohorts that the antibody response to the third dose tends to exceed the peak antibody response to the second dose. And that was true in our cohort as well. There was a significant augmentation of antibody responses after the booster compared to what we saw after dose 2. But the situation was actually quite different for the CD4 T cells. So although the CD4 response had waned over time, the third dose did do a really good job of bringing those CD4 T cell frequencies back up, but they reached frequencies that were quite comparable to what we saw after the peak of dose 2. So there didn't seem to be that sort of extra advantage of the third dose in terms of driving higher spike-specific T cell responses. We also tracked the kinetics of these T cells and antibodies in this cohort. We found that the T cell responses tend to peak in both frequency and activation by day 6 post-vaccination, at which point they started to rest down and decline in the blood. In contrast, the antibody response peaked between day 9 and day 12 and seemed to be generally more stable over time. One thing that was quite interesting with this cohort, which I didn't expect to see, was that the T cell responses that proliferated and activated in response to the third dose did not generally have a cTFH phenotype. So despite the fact that we saw robust antibody responses to this dose, there was actually very little evidence for the induction or the recall of a cTFH, spike-specific cTFH population in this cohort. And we can compare this to data that people have generated using HA-specific tetramers from influenza vaccination. And again, there's a stark contrast there, where typically 40 to 60% of those flu-specific cells do have a cTFH phenotype after annual influenza vaccination. So this was really quite a striking observation that we didn't expect to see. Now, having compared the CD4 responses in our infection cohort to our vaccination cohort, and we also wanted to ask about the combination of those two things, or the situation of hybrid immunity, as we often talk about it. So again, we tracked our convalescent cohort up to a year to 15 months after infection, and about half of that cohort was subsequently vaccinated with the first dose of a SARS-CoV-2 vaccine, in this case, either the AstraZeneca or the Pfizer vaccine. And just to put the boosting responses into context there, in terms of antibodies, the hybrid immunity cohort, so convalescence followed by vaccination, showed comparable neutralizing antibody titers to what we saw from our third dose vaccine cohort. So again, there was a really robust serological response. We were able to see robust expansion of our spike-specific CD4 population after both vaccines in the hybrid immunity cohort. And again, when we are tracking the kinetics of this response, we tended to see peak T cell responses occurring between day 9 and 11 in these cohorts. Interestingly, we also saw some heterogeneity and variability in the phenotype of these recalled CD4 T cells. So during the course of recall, around day 5 or so, we saw that the phenotypes diversified from that central memory phenotype that I discussed into a more heterogeneous mixture of effector, transition, and central memory. And we also saw a brief burst in CXCR5 positive T cells at this time point, which then returned more to the baseline characteristics of the tetramer cells that we saw following infection. Now, the one really striking feature of the hybrid immunity cohort that we didn't see within our third dose cohort was the induction of this activated cTFH response. And these plots will look quite similar or familiar to anyone who's familiar with influenza literature, where you see the appearance of these really activated CD38 ICOS double positive cTFH at about day 6 after vaccination. And these cells had a predominantly CXCR3 phenotype, again, really similar to the cTFH response that we see after flu vaccination. And studies of the flu vaccine have suggested that within this population, at least 40% of these activated cTFH are specific to the vaccine antigens. And in fact, using the spike-751 tetramer, we found that between 10 and 12% of these cTFH in some individuals had a single epitope specificity, which I think is quite interesting. The other really nice way that we were able to use the tetramer in this study was to track the phenotype of these activated cells longitudinally after vaccination. And in that case, we were able to see that although in the early days, they really were ICOS CD38 PD1 triple positive, they tended to rest down and lost expression of ICOS and CD38 relatively rapidly, but over time did continue to also lose expression of PD1. So by a month after vaccination, a number of these spike-specific cTFH cells were now PD1 negative, ICOS and CD38 negative, really appearing to just be components of the resting cTFH population in the circulation. And just finally, in the last couple of minutes, I just wanted to highlight some additional work that we did characterizing the TCR sequences of the cells recognizing this particular epitope. These are single cells that we single cell sorted and then TCR sequenced from our naive vaccination cohort. And you can see that there's really strong biases towards particular TCR alpha and beta chain pairings. And we saw very similar results in T cells that were sorted from our convalescent donors, both post-infection as well as post-vaccination. And across all 8 participants that we sequenced cells from, we identified a number of CDR3 beta motifs, as well as a number of public and private clonotypes. One of our public clonotypes was actually identified in 7 of the 8 individuals within the cohort. And interestingly, another public clonotype that we found had also been previously identified by another group who took a very different approach to identifying spike-specific cTFH responses through TCR sequencing, showing that these results have been identified in multiple populations. The other advantage of the single cell sorting and clonotype tracking was that it allowed us to look for clonotypes that were established by infection and then recalled by vaccination. So in our hybrid immunity cohort, we were able to show that in all of the individuals that we looked at, about 25% of the post-vaccination repertoire was shared from the post-infection repertoire, showing direct recall of those infection-established cells. Additionally, this gave us a nice opportunity to compare the clonotype composition of the cTFH and the CXCR5 negative CD4 memory pool. And here we found quite strikingly that all of the clonotypes that we observed within the cTFH pool were also derived from the CXCR5 negative memory pool, suggesting that there's a substantial overlap between those two pools, and there isn't necessarily selective recruitment of one TCR into either a cTFH or non-cTFH fate. So I've gone through a fair amount of data in a relatively short amount of time, but I think if we have take-home messages from the study, the first is that spike-specific CD4 T cells do persist in a very long-term memory pool, and this includes both a conventional CXCR5 negative memory subset, but also a cTFH subset. And these cells can be quite quickly mobilized upon antigen re-exposure in the context of infection or vaccination. We found that TCR recognition of this particular peptide MHC complex was similar across both infection and vaccination and was largely shared between both the CXCR5 negative and cTFH pools. And finally, we had that really striking recall of activated cTFH in our hybrid immunity cohort. And then while these cTFH emerged with this activated PD-1 positive phenotype, they did eventually persist in the blood with a more resting phenotype that didn't necessarily express PD-1. So with that, I just wanted to acknowledge everyone who participated in the study. A lot of this work was driven by Kathleen Ragg with our lab, also driven partially by Stephen Kent and Adam Wheatley. And I really want to thank the cohort participants who stuck with us for quite a while and quite a number of samples to produce the data for the study, as well as our funders. And with that, I'm happy to take any questions.
That's fantastic. Thank you very much indeed, Jennifer. That's a real whistle-stop tour of a huge amount of work that you've put in there. So thank you for that. Please do raise your hand if you have any questions, or you can type them into the chat if you prefer. While we're waiting for those questions and hands to be raised, Jennifer, you mentioned a little bit about the differences that you saw there between the vaccine cohorts and the natural infections and the different CXCR3 staining profiles. Could you maybe allude a little bit more on that and dig us a little bit deeper?
Yeah, absolutely. So after infection, we saw after infection, the phenotype of the tetramer cells was fairly evenly split between CXCR3 positive and CXCR3 negative cells. But over time, the memory cells actually became more enriched for CXCR3 expression. And that was sort of the predominant population that we saw at about 15 months. In contrast, the vaccine elicited cells tended to be more double negative, so less CXCR3 expression. Although in all cases with recall, we sort of got that burst of CXCR3 positive cells initially during recall that then shifted a bit. And so it's not clear if there's recall of a selective CXCR3 population or shifts in the chemokine receptor expression, but it was quite interesting and a little bit unexpected given some of the phenotypes that we had seen by the AIM assay.
Yeah, no, thank you. That's great. And then in regards to the actual epitopes that you were using, did you have any kind of additional thoughts in regards to maybe other cohorts of different HLA types and different epitopes that you were kind of interested in exploring?
Yeah, so we focused primarily on this epitope. We did also compare within the study the S236 epitope, which was restricted by the same HLA allele, and we found the 751 response was quite immunodominant compared to the other response. So there was definite heterogeneity within that pool. I think it would be really nice to do some comparisons with Paul Thomas's epitope and tetramer, just because those cells seem to have such a different phenotype, and they've had some really nice lymph node data, and it would be good to kind of get those within the same cohort to look across different phenotypes and dynamics.
Great, thank you. Antonio, you have a question?
Oh, yes. Hi, Jennifer. Antonio Bertoletti here. I want to follow the question about the phenotype of Jeremy, because so what are actually functional implications of the fact that a T follicular helper cell is CXCR3 negative? It means that it's not going to go on the site of the infection or in the lymph nodes, because I think that's my idea.
Yes, no, absolutely. It's a really intriguing point, and there's a lot of discussion around at what point in the germinal center reaction do the cells that we actually see in the blood exit the lymph node, and what population are they actually most related to in terms of GC or pre-GC TFH. So certainly when we, in mouse models and in some of our human data that we have, proper bona fide germinal center TFH don't express either CXCR3 or CCR6. Whether that's part of an exit program or something that's imprinted going into the blood, I think is still a bit of an open question. There's certainly mouse data that suggests that circulating TFH are derived from cells prior to their entry into the GC, that there's not actually an exit from the GC, and that could explain some of those chemokine receptor differences. But I think that's definitely still a gap in knowledge in terms of understanding where those cells are coming from and what that chemokine homing is actually doing in terms of function and trafficking.
Excellent. Great. Well, thank you very much indeed, Jennifer. Really appreciate your time and